Hi guys, and welcome to my first educational video, which I'm going to give a go at. I'm hoping to get a whiteboard to do this, but at the moment I'm going to be using a notepad. So, to begin with, we're going over a subject from Biology Additional, which is protein manufacture, or otherwise known as protein synthesis. Right, now remember, protein synthesis is when DNA is used to make the product which it codes for. So a muscle cell or something like that, known as a protein, which I'll be explaining to you. Hi guys, and welcome to my first educational video, which I'm going to give a go at. I'm hoping to get a whiteboard to do this, but at the moment I'm going to be using a notepad. So, to begin with, we're going over a subject from Biology Additional, which is protein manufacture, or otherwise known as protein synthesis. Right. Now, in protein synthesis, let me just explain to you what this is, in case you don't know. This is basically when you are creating new DNA using already made DNA. It's basically how the body basically produces new DNA. So, to begin this process, there are two sections. There is transcription, which is first, and then there is translation. So to begin transcription, this is the more simple one. But first, before I explain that, to remember the order between transcription and translation, remember that you write something before you translate it. That's how I remember it. You may obviously choose another method. So, for transcription, this is the most simplest one. So transcription begins with the DNA unzipping. So you have your DNA here, as you can see, and it's joined by bases. I apologise for my drawing, by the way. As you can see here. Now what happens during transcription is this DNA. So remember, you have your A and your T and your G and your C bases. So these bases will then break apart from their joining. This is done by an enzyme known as DNA heliocase. However, you probably won't need to write that in your exam. Okay, as you can see here, they break up. Now this strand of broken DNA over here, one of these strands, is known as mRNA. So I'll write that down for you. This section here, mRNA, and that's a little m. So this section of DNA is effectively half of your normal DNA. And the reason it breaks up is this small size allows it to exit in the nucleus, because otherwise it wouldn't be able to fit through and get to where it next needs to get to. Also, another important part to remember with transcription is that you have your 4 bases, and one of your bases turns into uracil, which is a different base. The base that turns into uracil is thymine, or T. So effectively, your T turns into a U. Okay, so that's another important section to remember. Okay, so let's just go over transcription again before I move on to translation. So, you begin with your DNA pairs here, joined up in a normal DNA as you would find it. And then it breaks up here as a result of an enzyme, and one of your strands is known as mRNA. And the T changes into a U with this mRNA. And that's just one strand of DNA, or mRNA, as I said. Okay, so translation. Translation is the second part of your protein synthesis. So, effectively, to begin translation, as I explained before, you will have your single structure of mRNA here, and these are bases that I'm drawing, these little rectangles, okay? They represent a base, okay? So, you have your mRNA, and this mRNA has just exited the nucleus, as I explained would happen. So, this mRNA will then enter a structure known as the ribosome. Okay? So, I'm just going to draw it as a square for now. So, as you can see, it will enter the ribosome and form here, into the ribosome. Okay, so it will exit, it will go into the ribosome here, and in the ribosome the DNA, or the mRNA, sorry, is decoded. What that means is this is a process where another type of DNA, known as tRNA, will join up to what is known as a codon. Okay, so a codon is a set of 3 bases. Okay, so you've got these 3 bases here. That is a codon. So your tRNA will come along. I will draw this on a separate page. What your tRNA is it will select 3 codons, okay? So you might have ATU here. So what the tRNA will do is it will select these 3 bases, known as a codon. Okay? And these 3 bases will be selected by the tRNA, but the tRNA will only select a certain codon, so a certain order of these 3 bases. So if this was, for instance, a G, this would not select it. And that's based on something known as an amino acid, okay? So an amino acid. Now this amino acid is something that's attached to the tRNA. So, you've got your tRNA here, and attached to it, it will have an amino acid, which I'll just put as AA for now, okay? So you'll have an amino acid attached to a tRNA. And what the amino acid will do is it will code for the tRNA to code for a specific codon. So it will tell the tRNA what codon it needs to select. Okay? So I will write that down for you. So, amino acid tells the tRNA what codon to attach to, okay? So the amino acid might code, for instance, for ATU, and therefore the tRNA will attach to ATU. Now, once the tRNA has attached to a specific codon, the amino acid which is attached to it also will be released. And once it's released, it will then begin to form a chain with the other amino acids that are released. So you will then have a chain of amino acids that begin to form because it will bond to the other amino acids that have been released. Okay? And this chain of amino acids is known as a polypeptide chain. Okay, so your amino acids will be released after the tRNA has attached to a specific codon or triplet base which the amino acid has coded for, and it will then form with other amino acids and bond to them to form a polypeptide chain. Now the last bit of our translation is simply this chain here will fold depending on how many amino acids it has got attached to it and what those amino acids are. So say there's 3 here, they might be different types of amino acids, and then that will cause it to fold in a certain way. Once an amino acid folds, so it will fold. So once an amino acid folds, it will then form a protein. So it folds to form a protein. And that protein is in a specific thing in the body. Might be part of a cell, might be a muscle, might be, I don't know, but that is what it will form. And then that's effectively how you create new things in your body using DNA. So let's just go through the process once more in a simple step diagram. So let's just go through this one more time simply in a step-by-step flow graph. So first off, your DNA unzips, okay, and that makes mRNA. Then the mRNA exits the nucleus. Because of its small size, it can do that. So remember, these first 2 steps here are known as transcription, yes? Okay, so after transcription, as I said, the mRNA exits the nucleus. Then, after the mRNA has exited the nucleus, it will enter the ribosome, which is outside the nucleus, okay? Then, the mRNA is read by the tRNA, and the specific amino acid chooses a specific codon. So a specific, remember, a codon is 3 bases in an order, yes? Okay? So then, the amino acid is released after it's attached to the codon, and it will then attach to other amino acids that have been released to form a polypeptide chain, and then, lastly, this chain will fold to make a protein. Thank you for listening. I hope you enjoy my video. This is my first time, so I'm not really used to doing educational videos, and I know it's just on a notepad, but I hope it's helped to explain some of the processes to you. Please include any recommendations or anything you want me to cover, in specifically biology. I'm focusing on B2 at the moment for EDEXEL, so anything you want me to cover, please just let me know and I'll see what I can do. But I hope this has explained to you more, and of course leave any questions in the comments and I'll see if I can answer them. Thank you very much for listening.